Hi, my name is Wendell Huang. Today is October 30, 2025. So today I'm going to present to you the essential roles of transition metals in biochemistry. It is one of the many essential elements that is critical to our body's functions. It can range from energy production to gene regulation. Now we're going to cover 13 objectives, which I did not write here because it's a little bit too much and too text heavy. So I'm going to focus on the main objectives. And to understand transition metals, we're going to focus on Lewis acid, Lewis acidity, multivalency, and structural motifs like zinc finger, and review clinical examples like sulfite oxidase deficiency. And finally, we'll take a look at how the body controls their uptake. Now, transition metals like iron, zinc, copper, molybdenum are essential micronutrients, meaning they are required in trace quantities, which is less than 0.01% of our body weight. They are indispensable prosthetic groups or cofactors for thousands of proteins. And imbalances like iron deficiency or genetic copper disorders lead to severe diseases because these metals are integral to core metabolic functions. Now let's talk about multivalency. If you ever study chemistry, you'll be aware there are different kinds of metal ions. And if you know that group 1 or group 2 metal ions, like the alkaline metals or alkaline earth metals, those are like stabilized into specific oxidation states, like +1 or +2. For example, sodium is +1, calcium is +2, almost all the time. Now, for transition metal ions, they can adopt multiple valence states. For example, iron can cycle between +2 and +3, which is the basis for their ability to be electron carriers in oxidation reduction, or aka redox reactions, such as those found in the electron transport chain. One thing I want to point out is that transition metals are considered Lewis acids. So that's a little bit different from Bronsted-Lowry acid because Bronsted-Lowry acid focuses on the movement of the proton ion, which is the hydrogen without the electrons in solution. But Lewis acid refers to something that can receive electrons, can accept an electron pair. And this makes transition metal able to enhance nucleophilicity of water, which is, it sounds like a very fancy word, but it just means that it makes water be attracted to a positive charge and turn water into a potent nucleophile. This is a central mechanism for enzymes that catalyze hydrolysis, like carbonic anhydrase, for example. The transition metals are rarely found free in the body. They are tightly sequestered in organometallic complexes bound by oxygen, nitrogen, or sulfur atoms from either like amino acids or as prosthetic groups. This complexation offers a lot of benefits. First, it ensures solubility and protects their functional oxidation state. For example, keeping iron stable in a hemoglobin. And it prevents them from randomly binding to anything they come in contact with, and you can control what it binds to. Now, complexation also suppress the generation of reactive oxygen species. By binding metal tightly, the body prevents free iron or copper from creating something called Fenton reactions, which is basically these metal ions creating reactive oxygen species like superoxide or hydrogen peroxide, which can damage cells at a really high level. Now, the surrounding ligands also tune the metal's redox potential, to match a specific enzymatic task. And complexes also allow the assembly of multi-metal units for cooperative chemistry, as seen here in this image with two nickel ions and urease. Now, iron is it's probably arguably the most versatile metal. Its function depends entirely on its protein environment. In hemoglobin, heme and iron is stabilized to act as an oxygen carrier. Conversely, in cytochromes, which is part of the electron transfer chain, which has a whole bunch of these iron sulfur clusters, iron is tuned to rapidly cycle between iron plus 2 ferrous and iron plus 3 ferric to serve as an electron carrier. Well, it even participates in non-redox reactions such as hydrolysis catalyzed by purple acid phosphatases. Now, copper is also very essential for many metal enzymes. It's critical for oxygen activation and catecholamine oxidases. It can cycle between copper plus 2 and copper plus 1 ion. These reactions produce important molecules like epinephrine and melanin. More dramatically, in lysooxidase, copper catalyzes the formation of chemical crosslinks in collagen and elastin, providing exceptional tensile strength in our connective tissue. Now, molybdenum is the core of the essential molybdenum cofactor. This metal is central to enzymes like xanthine oxidase and sulfite oxidase. Its unique value is the ability to cycle through three valence states, +6, +5, and +4. This allows it to receive two electrons from a substrate like sulfite and then sequentially transfer them one at a time to single electron carriers such as cytochrome C. Now the importance of molybdenum is highlighted by the severe genetic disorder, sulfite oxidase deficiency. This is caused by mutations in genes necessary for molybdenum synthesis. Without functional sulfite oxidase, the toxic sulfite generated from the amino acid breakdown, such as cysteine and methionine, accumulates. This leads to profound physical deformities, such as brain damage, intractable seizures, and in most cases, death in early childhood. Now let's talk a little about zinc, because it's pretty unique because it's even in its +2 ion state, it's actually redox inert, which means that it does not change oxidation state, at least not very easily. This stability makes it the ideal candidate for two roles. First of all, it's really good for structural stabilization. The zinc ion is a prerequisite to stabilize the conformation of zinc finger proteins, ensuring they are correctly folded. This stabilization is critical for zinc finger motif and transcription factors. More importantly, contemporary research confirms that zinc finger proteins, due to their structural stability and flexibility, play pivotal roles in DNA damage repair, or DDR for short. You can think of them like as guardians of genome stability, and maybe serving as a potential new target for cancer therapy. Now, the second role of zinc is Lewis acid catalysis. Enzymes like beta-lactamase 2 use zinc +2 ion to simultaneously polarize the substrate and coordinate a water molecule, creating the necessary nucleophilic hydroxide to cleave the target bond, which is shown here in this image. This mechanism is central to hydrolytic enzymes like carbonic anhydrase and carboxypeptidase A. Now, unlike the essential metals we just discussed, toxic heavy metals also exert damage on the body, despite the usefulness of many of the metals. First, there's cation displacement. So they mimic essential ions like cadmium 2 displacing zinc 2, which inactivates essential enzymes. Second, there's also enzyme inactivation. They bind to the reactive sulfohydryl groups of cysteine residues and permanently inactivate the enzyme. Third is catalysis of reactive oxygen species. It's similar to the misregulated iron and copper ions, and this can catalyze the Fenton reaction we talked about earlier, which produce a very destructive reactive oxygen species, like the hydroxyl group with that electron dot radical, hydroxyl radical. Our latest source also confirms that reactive oxygen species induce oxidative stress, which is a key feature in conditions like Alzheimer's, Parkinson's, or Huntington diseases. Now, treating acute heavy metal poisoning, you have to focus on removal and neutralization. The primary method is chelation therapy, where small molecules, chelators, are administered to tightly bind a toxic metal, rendering it inert and facilitating its excretion. Just pass it back out. Other supportive treatments include high water intake with diuretics and in severe cases, hemodialysis. Now let's talk a little bit about iron and copper absorption. Intestinal uptake of these two ions is highly regulated. So iron and copper uptake involves two steps. First, it's reduced by the surface enzyme DYCTB, or it's also known as duodenyl cytochrome B. The iron is then taken up by the general DMT1 transporter, which stands for divalent metal transporter. It's divalent because it tends to take the ions that have a +2 charge. Of course, copper ions also have their own respective transporter, which is the CTR1, or in other words, copper transporter, which is actually dependent on duodenyl cytochrome B. Now, divalent metal transporter is critical for iron absorption. It is also the primary vehicle for the uptake of other divalent metals. For example, manganese or nickel. This shared pathway is a key regulatory point, as excess zinc can actually inhibit copper absorption, which illustrates competitive uptake mechanisms. The absorption of molybdenum and vanadium is structurally driven because they exist in the gut as oxyanions, molybdate, and vanadate. They are absorbed by non-specific anion transporters. These are the same transporters responsible for absorbing their structural analogs, such as phosphate and sulfate. Finally, cobalt is absorbed by a totally dedicated pathway, since its sole function is part of vitamin B12, cobalamin. It is absorbed only as the organometallic complex. This requires 3 specific proteins: haptocorin in the stomach, intrinsic factor which binds to it in the duodenum, and finally, cubalin receptor which mediates the uptake in the intestinal cells. Okay, let's do some review questions. So, which transition metal property is described as its ability to act as an electron pair acceptor, which is harnessed in catalysis to enhance chemical reactivity, and is exemplified by the activation of water molecule by zinc ion? All right, the correct answer is Lewis acidity. I mean, the definition of Lewis acid is basically an electron pair acceptor. This property is vital for many enzymes, such as carbonic anhydrase. By accepting this electron, the metal actually polarizes a nearby molecule, in this case, turning water into a highly reactive nucleophile. Unlike iron, Which cycles between Fe2 plus and Fe3 plus for electron transport? Zinc is described as being redox inert. What is a key structural role that's this stability of zinc 2 ion make it ideal for, according to the text? So the answer is stabilizing the conformation of zinc finger proteins, such as the zinc finger motif and transcription factors. Because zinc plus 2 ion is redox inert, it means that it doesn't really change its oxidation state. It is perfectly suited for such structural role. It can act as some kind of anchor, stabilizing the folding of certain proteins, specifically the zinc finger motif in transition factor. This is also what's important for DNA for proteins to accurately bind to DNA. The body has tightly sequestered transition metals and organometallic complexes. Which of the following is not listed in the text as one of the 4 vital benefits of this complexation? The answer is facilitating the excretion of the metal from the body via chelation. Now, chelation is a very helpful therapeutic technique nowadays to remove toxic metals, but our body doesn't actually naturally do it. So that one is not really included. Cobalt absorption is unique because its sole function is part of vitamin B12, cobalamin. What is the correct sequence of the 3 specific proteins required for its dedicated uptake pathway? Haptocorin in the stomach, intrinsic factor duodenum, and tubulin receptor for intestinal cell uptake. Pretty straightforward. The toxic heavy metals exert their damage through 3 primary mechanisms. Which of the following describes the mechanism of enzyme inactivation by these metals? They bind to the reactive sulfohydryl groups of cysteine residues, permanently inactivating the enzyme. Now, toxic metals are chemically attracted to sulfur atoms in the SH groups of cysteine. So by binding tightly to these residues, they distort the enzyme's active site and pretty much permanently shutting down its function. Options A and D are also toxic, like mechanisms of toxicity, but they are more related to cation displacement and catalysis of reactive oxygen species. Now, here are some key takeaways here. To conclude, transition metals are indispensable for their multivalency, which enable electron flow and redox reactions, and there's Lewis acidity, which enable powerful hydrolysis. And they must be packaged via complexation to prevent toxicity and enzyme specificity so that it targets the specific molecule it's designed for. And their intestinal uptake is regulated by diverse, specialized, and often shared transporters. Thank you. I hope you liked this presentation.